Hi, I'm Haley, and today I'll be talking about experimental evolution in yeast. The two species I'm focusing on are Saccharomyces uvarum and Saccharomyces cerevisiae. We decided to investigate the differences between these two species using experimental evolution. Experimental evolution has been used as far back as 1887 with Dallinger over here, and as recently as today with Richard Lenski's long-term E. coli experimental evolution project. Cerevisiae and uvarum are closely related species that have diverged in their thermal growth profiles. Cerevisiae and Paradoxus grow well in warmer temperatures, whereas uvarum grows well in cooler temperatures. The aims I'll discuss in this presentation are: determine whether temperature-sensitive species can adapt to high temperatures, and if so, by many small steps, few large steps, or something in between, and determine whether high temperature adaptation has a cost at low temperature. These are the strains that we used in the experiment and the abbreviations I'll be using for the rest of the presentation. We have our heat-tolerant Cerevisiae and Paradoxus and heat-sensitive uvarum, as well as 5 hybrid strains for aims not discussed in this presentation. So we have our 8 strains, and we have 48 replicates of each strain for a total of 384 positions per plate. Then we want to put 3 different temperature conditions, so we have to make 3 copies of this plate using a process called replica plating. Each of those 3 plates goes into one of the temperature treatments for a total of 1,152 evolving lineages. Now that's a lot of evolving lineages, so it wouldn't have been feasible for us to do this experiment without the robot. Here's a little video of the robot replica plating. So we run into a little bit of a problem. We want to evolve uvarum to 37 degrees, but that's a lethal temperature to them if we kept them at 37 the whole time. To solve this, we decided to switch off between being at 37 degrees and recovering at a more comfortable temperature for them. Throughout the experiment, we monitored our progress, or more like the yeast's progress, by taking pictures of the plates in each cycle of treatment and recovery. We took pictures using the PhenoBooth, which is able to calculate the size of each of these positions on the plate. More on that later. So here's the big picture. We have a plate. We put it in treatment for 24 hours, one of those 3 temperatures. Then we allow it to recover at a comfortable temperature for 48 hours. We take a picture of the plate using the PhenoBooth. And then we make a copy of it using the robot and start the process anew. So now let's look at the data from those pictures that we took throughout the experiment. We have size over time for our 3 temperature treatments, and we can see that in all of them, there's a general trend of improvement over time. And if you see uvarum in pink, in the 37 treatment, it's definitely improving over time, although only by a little bit, suggesting maybe by many small steps. So this is great, but it doesn't differentiate between physiological adaptation and genetic adaptation. That is, the cells could look like they're adapting, but actually just be physiologically prepared for the treatment. To address this, we did a phenotype assay where we took the evolved plates and we froze them, essentially cleaning the physiological slate, and ancestor plates, we also froze those. Then we put them through a cycle of treatment and compared the resulting sizes. Now we're going to look at that data. In the following graphs, dots above the line mean that the evolved lineage is doing better than the ancestor, whereas dots below the line mean the evolved lineage is doing worse than the ancestor. On this graph, we have the lineages evolved at 37 degrees compared to the ancestors when tested at 37 degrees. If the uvarum had only physiologically prepared for the treatment, we would expect about an even spread on either side of the line. If, however, they had genetically adapted to the temperature, we would expect more dots to be above the line than below the line. If they adapted with big leaps, we would expect those dots to be far above the line. If, however, they were using small steps, they should be just barely above the line. And what we see is indeed all these pink dots, the uvarum, are above the line, showing that they did genetically adapt, and they're just barely above the line, showing that they used small steps. So the uvarum adapted, but did it adapt to the temperature treatment or just to the media that we were using? To figure that out, we looked at the lineages evolved at 37 compared to the ancestors when tested at 20 degrees. If they were evolving to the media, we would expect that they would be just as equally better than the ancestors at 20 degrees as they were at 37 degrees. If, however, the improvement over the ancestor was temperature specific, we would expect them not to do any better than the ancestors when tested at 20 degrees. And what we see is they don't do any better than the ancestors at 20 degrees, showing that the uvarum was adapting to the temperature treatment, not just to the media. Now we can address aim 2, where we want to know whether there's a cost to this adaptation at high temperature. We look at the lineages evolved at 37 and see how do they fare at 4 degrees compared to their ancestors. If there was a trade-off, we would expect more of the pink dots to be below the line than above the line. But that's not what we see. We see the dots pretty evenly spread on either side of the line, showing that there was no obvious trade-off at 4 degrees. So we figured out that yes, temperature-sensitive species can adapt to high temperatures by many small steps, and no, this does not necessarily incur a trade-off. I would like to thank Emery, Su-Ying, Nalima, and Justin, and a special thank you to Tiffany and James, without whom this project would not have been possible. And thank you all for watching.